Hello, my name is Brandon Kovarovic. This is a overview of my PhD research in the Biofluids Research Group of Stony Brook University in the Department of Biomedical Engineering under the advisorship of Danny Bluestein. My research has focused on developing patient-specific in vitro models, which were then adapted into in silico models for evaluating transcatheter aortic valve clinical complications. So the pathology that we're treating is calcific aortic valve disease, which is characterized by a progression of calcific masses growing on the aortic valve. These masses grow in size and become more rigid over time, causing a condition called aortic stenosis, which is a increase of the transvalvular gradient, restricting the opening of the aortic valve and putting a burden on the left ventricle, which, if untreated, eventually leads to heart failure. The traditional therapy is a surgical valve replacement in which the patient receives open heart surgery, the stenotic valve is dissected out, and a new valve is sutured in place. This is a highly invasive procedure, and it's estimated that 20% of these patients that should receive a surgical valve do not because they are deemed too frail for operation. So this is where transcatheter aortic valve comes in. The valve is replaced in a transcatheter technique. You can see in this animation here from Edwards. The valve is guided up from a delivery catheter from the femoral artery. It is placed inside the aortic valve and inflated either via balloon or with the stent frame, crushing the native valve into the walls and placing the functionality of the valve with a new valve. What's important to note here is that in the transcatheter technique, the native valve is not removed and these calcific masses remain. These masses have a characteristic patient-specific distribution and size that varies with each patient. TAVR has been life-changing, saved many patients so far, but it does come with complications, and these complications generally mean worse outcomes for the patient. These complications include paravalvular leak, which we are going to be addressing. These are small gaps that occur between the stent frame and the native lumen, causing regurgitant flow back into the ventricle. These are small little jets, and the diastolic gradient across these jets it's the most extreme gradient in the vasculature. So the jet flow is pretty high, even though these are small gaps. Patient prosthesis mismatch means that the valve is supposed to perform at a certain rate and it winds up having a poorer performance, lesser performance, and this is generally associated with not matching the right size of valve to the patient. Thrombosis is becoming an increasing problem where actual activated platelets are depositing either on the leaflets or in between the leaflets and the native sinus, which then can lead to stroke. Other complications, older ones, would be device migration. You can see in this video, the sapien valve had actually migrated into the ventricle of the patient. And finally, there's an increase in pacemaker implementation with TAVR as the stent frame is placing stress on the AV node or the LVB region. So for testing aortic valves, we use a benchmark called the ISO 5840-3. This ISO standard is for evaluating both the hydrodynamic performance of the valve, both the systolic and diastolic performance, as well as the durability of the valve over time. I'm going to be just addressing the hydrodynamic testing, which is how you would benchmark the valve against other valves and really evaluate its pluses and minuses. So traditionally this is done in a machine like this called the in-vitro left heart simulator. And we are referring to it as an idealized geometry because you'll see that the aortic structure is straight and there are no native valve leaflets or calcifications present to actually distort and deform the stent frame. So from my research and working with this local company in Stony Brook University called Vascular Simulations, we came up with a patient-specific simulator called the Replicator Pro, seen here. It contains all the major vessels, all the major arteries inside the body, as well as realistic and physiological flows, including control over the cardiac output, the blood pressure, and independent control of all the flows throughout the anatomy. It offers a pretty unique platform in which we can simulate an entire procedure on what we deem as a patient, even though it's a plastic and silicone patient. It contains a functional left heart. The heart is actuated pneumatically, and everything is controlled through an adaptive feedback mechanism. So you can see that these two structures are completely different. One actually has a native valve with calcifications. The other one has no valvular anatomy, in which the actual device is just deployed against a silicone cylinder. And the valve is actually, or the aorta is just a perfect straight tube, as opposed to an actual patient anatomy. The interaction of these components actually define how the valve is going to work inside the body. And if we look at just these pressure and flow waveforms between the two machines, we can see that there is an increased gradient as the deployed valve is more stenotic. So this offers a nice complementary platform to study valves against, whereas the deformation of the valve will be more realistic to what we would see in the clinic. And we can actually estimate kind of the clinical parameters of the valve prior to even doing a clinical trial, just by mimicking the functionality and geometry of the patient and testing for these different conditions. So that being said, for this study, we chose 5 patients. The patients' CTs were segmented, and a design and an actual model was produced. The models are made out of a polyurethane elastomer with semi-rigid calcific masses that are actually affixed to the surface of it. These masses can be colored with a radiopaque material if we want to evaluate how it looks under fluoroscopy. And they can be swapped out and changed inside this replicator system repeatedly to evaluate different valves in sequence. These are the 5 patients chosen. Each one has a different condition that we chose, and they were all scaled in order to give the same oversizing of the valve that we're testing. It's also important to note here that I'm testing both calcified and non-calcified models in order to distinguish what the difference is between the calcifications and the interaction of those stents. So in this study, we're comparing a new novel polymeric TAVR valve that we're developing in the lab in conjunction with Polynova Cardiovascular against a Brazilian tissue valve. It has a completely different stent design with a balloon expandable steel frame versus the self-expandable nitinol frame. So we're not only evaluating the differences in the leaflet technologies, but also the stent technologies and comparing the two. Our goal is to characterize the hydrodynamic performance between these two valves and kind of benchmark them against the native valve, because we now have that ability. All these measurements will follow the ISO standard, so they will be directly comparable to the idealized conditions. For systolic performance, we compare what's called an effective orifice area. It's basically a measure of how opened the valve is during systole, and also an average pressure drop across the valve. For diastolic performance, we evaluate the closing flow as well as the leak flows, and then the combination of the two we refer to as the total regurgitant fraction. These are going to be recorded at 70 BPM at normotensive conditions and at 4 different cardiac outputs in order to give a range that you'd see inside of a patient. So you go through, you make the models. First thing is you record the actual replica. So we're getting a benchmark here of the native stenotic replica. Then you deploy the different technologies inside. These are just simple endoscope videos that we took from the patient anatomies. You can see the difference between the valves. You repeat them for the other remaining anatomies, and then you can actually test them in the idealized conditions. You could see in the idealized simulator, it's nice smooth video, high-speed images, which you can attain because it's a perfectly straight aorta, and it's very easy to align an actual camera to it. One of the immediate things you'll notice here is that the idealized conditions, the polynova valve highlighted in blue, is perfectly circular compared to the patient anatomy, where it's actually eccentric and elliptical, and the same goes for the tissue valve as well. So you can get a more realistic scenario of the native valve interaction inside these patient anatomies compared to the idealized anatomy. So observing the forward flow performance, so the y-axis here is the effective orifice area. And it's important to note that all the valves have passed the ISO standard. It's a fairly relaxed standard and wants to pass valves through in order to be evaluated clinically. So the goal here is to distinguish the performance and distinguish the benefits of each valve more directly and what that actually means inside the clinic, as opposed to they all perform equivalent, which is what the ISO standard is basically saying inside the idealized conditions. So the first thing you'll notice is that it's very dependent on the anatomy, how the performance occurs. The two different devices had distinct performance in 8 out of the 11 cases, with the polynova performing lower in the highly stenotic anatomies, and thus making the performance distinction between the two valves less. And then the major finding here is that the idealized case is actually overestimating the performance of the polynova valve in 7 out of the 10 cases, and the Innovair valve 3 out of the 10 cases. So if you were to just follow the ISO standard, the polynova polymeric valve would look amazing and should perform the best, but in actual realistic scenarios, we learned that we should not expect this in the clinic, and we can actually more refine the design in this stage of the process rather than go back, go to the clinical trials and then have to repeat everything all over again in order to drive even more distinction and innovation. The native valve is linked to the performance of the valve, so the more stenotic the valve, the more stenotic the TAVR valve, which is expected, but it's interesting to confirm that. In terms of the leaks, we're looking at just the leak flow here on the y-axis. Again, highly dependent. The polynova has no anti-PVL features, so no sealing, skirt, or anything like that. But it is, in some cases, it's able to, the nitinol frame actually allows it to adapt to the native lumen better and seal better than the Innovair. But if we want to look at the distinction between the non-calcified and calcified valves, if we look at patient C, you can see that there's a great discrepancy between the calcified valves and the non-calcified. And if we look in the actual anatomy, we can see that the valve is deployed against these annular calcifications. So the leak path is actually occurring because of these annular calcifications, and we can actually show that and prove that in this model. One of the benefits of the replicator system is that it can basically be viewed as a patient. All the hydrodynamic performance testing that you saw previously was all done on the benchtop, on a cath lab table with an ARTIS Zigo. And with this system, we can actually do a intraprocedural CT. So the replicator was rapid paced to 180 BPM. And the blood pressure was dropped down to 20/10 millimeters of mercury and allowed to just pace back and forth for approximately 10 seconds. A 5-second spin is, you could see here, to take one scan, and then contrast is injected and you could take another scan. So if we look at the initial scan, we can actually determine and reconstruct the stent geometry and where it aligns inside of the lumen. This gives us the ability to reconstruct the stent and actually measure its deformation, and we can actually correlate that to the performance characteristics, showing that the variation in the polynova performance is directly linked to the variation in the stent. From a design standpoint, we can go back and say that the stent geometry needs to be refined in order to have a more consistent deployment against these patient-specific anatomies. So that means either refined stent structure or a more rigid frame, maybe adapting from the cobalt chromium frame going to a balloon expandable valve. The point is, during this process, we now know how it's going to benchmark inside the patient, and we can now adapt and change it. So with the contrast injection, we can basically do a subtracted scan and get the geometry of the contrast. And then if you look here, we can appreciate the shape of the PVL flows that occur between the stent frame and the native lumen. You can actually resolve it. And then lastly, we could see that you're getting leak flow in that model with the annular calcifications, thus proving or giving more evidence that the leak flow was between those annular calcifications. So now that we've established that there is a large discrepancy between the idealized models of the current ISO standard and what is done in industry currently and what could be done with patient-specific models, we can take this research to the next step and actually evaluate it with in silico measurements for greater detail and fidelity. So taking these hydrodynamic measurements that we've shown previously, we can take that model out of the replicator, place it into a micro CT with a custom jig doped with contrast so that we can actually resolve the features of the native leaflets versus the TAVR leaflets, and then painstakingly reconstruct it into 3D geometries. These geometries are then meshed into a CFD domain, giving us a natural ability of validation given the conditions and the measurements done in vitro are then taken in silico. So making a CFD model from this, basically we're using ANSYS fluent in a laminar flow solution with polyhedral cells, between 3 and 3.5 million cells. And we're taking those boundary conditions from the in vitro study, extending the geometries and applying them to the ventricle and aorta. And then the goal here is to study the 5 patient models with the polynova, determine what the associated thrombogenic potential from the shear stresses of these PVL flows are. So we do that with a DPM model that seed Lagrangian platelet-like particles. These particles, between 2 and 3 million particles, are seeded during one cycle of systole and then are allowed to wash through the domain for the preceding 3 cycles, and they're continuously tracked. So one of the novel things about this study is I basically varied the permeability of the leaflets, allowing me to capture both systolic and diastolic flows. So during systole, the leaflets are open and permeable, allowing the gradient to force forward flow through the valve. And then during diastole and closing flow, it becomes gradually less and less permeable and thus is closed. This gives me the ability to mimic the bulk flow of a valve without resorting to fluid structure interactions, which would complicate the system even greater. But this gives me an excellent match between the in vitro results and the CFD results, keeping in mind that there's a large error associated with the flow sensor. Here on the left, you see some volume renderings of the flows, and you can actually see the PVL flows on the side during mid-diastole. So with the particle seeding, we can first see the first evidence shown that there are particles that enter the PVL gaps during systole. It's a far lower gradient during systole, anywhere from 10 millimeters of mercury up to 40 millimeters of mercury in my models. And it's a shorter duration, so the likelihood of particle of platelets entering that domain is less. But with this model, we can see that there is still a likelihood. And then we can see actual realization of the particles flowing through, washing back into the ventricle, and then reincorporating into the systolic flow. The number of particles appears to not be dependent on the leak flow and highly dependent on the actual jet velocity, with the more stenotic valves causing more platelets to jettison downstream, down to the descending aorta, as opposed to remaining in the ascending aorta and native sinuses to then wash through the PVL flows. Here you can see two animations of the particles in the first anatomy. The left shows the particles colored by velocity, and the right shows them colored by stresses. On the right, you can start to appreciate the actual stresses associated with this and the velocities associated. So with a higher velocity in the PVL gaps, you also get higher shear stresses and thus higher stress accumulations. So what we do in our lab is determine a thrombogenic potential from a stress accumulation tensor. This is basically the average stress on the particle tracked over time and accumulated throughout its trajectory. From this, we determine a probability density function. So in this model, I'm able to distinguish between the particles that entered the PVL domain versus those that didn't. And we can clearly see that because the PVL particles are shifted to the right, they have much higher potential stress accumulations, thus a greater increased thrombogenic potential associated with them. This is also the first study of evidence that particles have a tendency or a likelihood of reincorporating into PVL flows. So what this means is, given the 3 cycles, they're able to enter either in systole or diastole. And I've shown that some particles are entering systole and diastole in each cycle up to 6 times. A very small amount of particles are able to do this, but they're basically washing either from the ventricle into the native sinuses and back through many times, or they're going through in systole and then washing back and forth. Showing that the closer they are to the native lumen, the more likely they are to be incorporated into the PVL flows. And then with that is associated a greater increase in stress accumulation. So the more times they pass through it, the greater thrombogenic potential they have, the greater stress accumulation they have. So in conclusion, we can show that there, again, is an overestimation with the idealized geometries. We can characterize the TAVRs as if they were in the clinic, thus decreasing the likelihood of complications in trials, or at least giving us the opportunity to mitigate these complications before the trials, reducing patient risk. I can use this patient simulator as like a guinea pig for novel imaging, as you saw with the intraprocedural CT. And then finally, with the in silico studies, I have the ability to actually validate them and to show an associated shear stress profiles with the PVL gaps. Another important aspect of this study is that all these valves have passed the ISO standard. Therefore, those leak patterns and associated shear stresses are things that would generally be ignored in the clinic, would be rated as mild PVL flows or trace PVL flows, and would generally be ignored, and the patient would be deemed as fine. So I'm showing here that even these smaller complications are more varied and can still result in things like stroke and thrombogenic responses. So with that, I would like to thank you for listening to my presentation and my research. If you have any questions, please feel free to contact me at brandon.kovarovic@stonybrook.edu. I look forward to talking to you. Thank you.